Hello everyone, welcome back to Python for Bioinformatics. In the previous video, we learned how to change the case of a DNA sequence. Today, let's look at a very common question when working with sequences. Does a particular nucleotide or pattern exist inside our sequence? Suppose I have this DNA sequence and I'm interested in the pattern GGA. I want to know whether GGA occurs somewhere inside this sequence. So how can we ask this question to Python? Now before we write any Python, think about what we are actually asking. I am not asking where GGA occurs, and I am also not asking how many times it occurs. I am simply asking, is GGA somewhere inside this sequence? So the answer can only be a yes or a no. And Python gives us a very simple way to ask exactly this type of question. The in operator. The general syntax is item in sequence. We put the things we are looking for on the left, in is in the middle, and the sequence we are searching inside on the right. And the result is a Boolean value. True if the item is present and false if it is not present. Now let's try it with our DNA sequence. Here I have stored the DNA sequence in the variable named as DNA. And now I am writing GGA in DNA. So here GGA is the pattern I am searching for, and DNA is the sequence I am searching inside. So Python is essentially asking, does the string GGA occur anywhere inside the string stored in DNA? And when I run this, it returns true. That means the pattern GGA is present somewhere in our DNA sequence. And if we look at our DNA sequence, we can actually see that GGA is present in it. So we have answered our original biological question. Yes, this sequence contains the pattern GGA. Now let's search for a pattern that is not present. I'll use CCC in DNA. Again, Python checks the entire DNA sequence to see whether this exact pattern occurs anywhere inside it. This time the output is false. In plain language, this means Python could not find CCC anywhere in the sequence. So in gives us a very straightforward yes or no answer. Now we don't always have to search only for multi-nucleotide pattern. We can also use in to check for a single nucleotide. And here I am asking three questions. Does this DNA contains A? Does it contain T? And does it contain N? These are all same type of question. Is this particular character present somewhere inside the DNA sequence? Now let's run these three checks. The first expression checks for A and Python returns true, meaning at least one A occurs in the sequence. The second checks for T, and again Python returns true. So T is present. The third checks for N, and Python returns false. So this means there is no N inside our DNA sequence. Now notice something important here. We are only checking whether the nucleotide exists. We are not counting how many times it occurs. Now this is an important limitation to understand. If I write A in DNA, Python is answering one specific question. Does A exist somewhere in the sequence? It is not telling us how many A's are there, and it is also not telling us where the A occurs inside the sequence. So when you use in, think of it as a presence or absence question. Is it there or isn't it? Now let's apply the same idea to a slightly more realistic sequence pattern. Here I have another DNA sequence and I'm looking for the 3 nucleotide pattern TAG. And when we run it, Python returns true. So we know that TAG occurs somewhere in this sequence. Notice that Python is checking the complete TAG. We are not asking whether T or A or G individually exist. We are asking whether those 3 characters occur together in that exact order. There's one more important thing to remember. When searching biological sequences, uppercase and lowercase are different characters. For example, ATG and ATG are not considered the same string. And this matters because our DNA sequence here is written in uppercase. Now when I search for uppercase ATG, Python returns true because that uppercase pattern occurs in our DNA sequence. But when I search for lowercase ATG, Python returns false. The reason is that Python is looking for the lowercase characters specifically, and those characters don't occur in our uppercase DNA sequence. So when working with sequence data, keeping the case consistent can be important. Now it's your turn. Here we have a RNA sequence and I want to know whether it contains the pattern UAC or not. Pause the video and try to write the Python expression using the in operator. Remember the structure. The thing you are looking for goes on the left and the sequence you are searching goes on the right. So the answer for UAC in RNA is true. This means that UAC occurs somewhere in our RNA sequence. Now let's quickly summarize. When we want to know whether something is present inside a DNA or RNA sequence, we can use Python's in operator. The basic syntax is item in sequence, and Python then gives true if the item is present and false if it isn't. The key thing to remember is that in tells us whether something exists. It doesn't tell us where it occurs or how many times it occurs. That's it for today's video. Thanks for watching. In the next video, we'll take this one step further. Instead of simply asking whether a pattern exists or not, we'll learn how to find its position inside the sequence using the find method.